Hello students, welcome to the class, welcome to the biology class students. Students, hope you all are well today. Students, one thing I would like to tell you about, there is no alternative to success students. It is a product of your hard work, it is a product of learning from the failures. Right students, so this is the right time students, get up and do the hard work until you succeed. So students, in the biology class, we were discussing the chapter reproductive health. Right, in this class only we will be discussing some basic patterns or some questions which are important as per your CBSE is concerned. So students, to talk about these questions, let me first brief you about the reproductive health. In this chapter we have discussed about, first of all, you should know what do you mean by this title, that is reproductive health. Reproductive health, as per WHO, that is the World Health Organization, it says that a person is considered reproductively healthy if it is healthy in terms of the social point of view, if the person is healthy in terms of the physical point of view, if it is healthy as per the emotional point of view. In terms of reproductive health is concerned, that person is considered as a reproductive health. Right. In this chapter, we have already discussed some of the reproductive strategies. Those are certain acts were there. We have discussed the problem which is related to the overpopulation. We have discussed the problem which is related to the sex education. Right, students? In this chapter also, we have already discussed that how we can control the population, or what are the how we can study the population, how we can calculate or study the demography. Demography is a study of population. Right, students? Hope you all know about this. So in this chapter only we have already discussed certain sexually transmitted disease as well. So to talk about the sexually transmitted disease, there are so many types of sexually transmitted disease that transfer from one particular organism to another with the help of physical contact or with the help of the sexual contact. That's why it is given sexually transmitted disease, right? In this chapter itself, we have already discussed some of the methods, right? Well, for the contraception, some contraceptive methods we have already discussed, some natural methods we have already discussed. We have already discussed some of the barrier methods like condoms, cervical caps are there. We have already discussed there are certain methods like hormones are there and some implants are there, or some external things are implanted into the body of a female or into the male, and some surgical methods are there due to which it leads to the contraception, or these are the methods of contraception, right? So students, to talk about in this chapter, there is one important topic, that is in vitro fertilization, or that is also called as assisted reproductive technologies. In the assisted reproductive technologies, there is IVF, that is in vitro fertilization. We have talked about the surrogacy. We have already talked about the artificial insemination, ZIFT, that is a zygote intrafallopian transfer, the gamete intrafallopian transfer. There are so many assisted technologies are there by which a couple can have the baby when those couple, they are unable to produce one baby. So with the help of these technologies, they can have the babies, right? So to talk about the certain questions which are related to the CBSE pattern, or which are important as per your CBSE is concerned, the first question is, yes students, the first question is what are MTPs and under what conditions MTP are legally permitted? Now the question arises, student, what do you mean by MTP? MTP, medical termination of pregnancy. So students, this MTP means medical termination of pregnancy, right? So students, as the name indicates, medical termination of pregnancy, that means to terminate a baby, right? And it is a voluntary action, that means a baby is terminated before the birth. Now the question arises, why there is termination? There are so many reasons for the termination. One of the reasons could be the failure of contraception, right? If there is a failure of contraception, there are chances that the people or the mother will have a congenital deformed baby. Right? So to avoid that, a couple go for this medical termination of pregnancy, right? If we talk about the general, that is, it is legalized, or you can say it is safe up to 12 weeks, right? Safe, they are safe up to 12 weeks, students. Right? It is safe up to 12 weeks, right? Because after this, what actually happens is that it is unsafe for baby as well as for the mother. So, if we talk about the total medical termination, the 20% of the total birth is terminated every year. And to talk about more in detail, 30 to 40 millions of pregnancy is terminated every year worldwide, right? To talk about in detail, that is, normally many countries, they do not have any kind of rule which are associated with this medical termination of pregnancy, because people feels that there are so many issues which are related to the medical termination, and that could be the social issue, that could be the emotional issue, that could be the religious issue. Because of so many issues, it is not legalized in many countries, but it is legalized in India, or some acts are there which are related to the medical termination of pregnancy. So, students, come at this side first. The first act which was passed is the Medical Termination of Pregnancy Act. Medical Termination. Students, is this visible to you? Of pregnancy. And this act was passed in 1971, right? And later on, so many amendments are done in this particular field only. Now the question arises, why this particular act came into existence? Why this is an act in 1971. So question arises, why this came into the scene? Why? Just because there are so many death, like total, if we talk about 8.9% students, 8.9% of the total death, right, of total maternal deaths, right, 8.9% of the total maternal death in India is because of this unsafe pregnancy, right? Of this maternal death is due to unsafe pregnancy. So it's very important to combat with such type of problems. So this act came into existence, right? And later on, one more act that came into existence, that was pre-diagnostic test regulation and uses. That was a prenatal diagnostic test act, right? So later on, another test came into the existence. This was the Prenatal Diagnostic Test Act. So students, the question arises, what was the use of this prenatal diagnostic test? Normally the problem in India is the female feticide. Female feticide. Female feticide, right? The major problem in India is the female feticide. Like, why people go for this particular procedure, the female feticide? With the help of certain procedures are there. There are amniocentesis, and another is the sonography, right? To talk about this, there are two procedures, students. Amniocentesis and second is a sonography. These are the two tests by which we can get to know whether the child is having certain malfunctions or some congenital disease are there in the child or not. So we get to know whether the child is having congenital disease. Right. So actual use of this is amniocentesis and sonography is to check whether the child is having this congenital disease or not, right? But nowadays what happens is, with the help of these amniocentesis and sonography, we are misusing this particular procedure, and we get to know whether the child is a female or whether the child is a male. So because of some mental issues in those people, they go for the female feticide because they want a male child or they want a boy, right? And now you'll talk about what do you mean by amniocentesis. Amniocentesis is a procedure, because I have used this term, so it is a right point to tell you about the amniocentesis, right? Amniocentesis is a procedure by which we take out the amniotic cells. Now what happened? Now you'll ask, what do you mean by amniotic sac and amniotic cells? Baby is formed, or baby is there in a one particular sac, and that sac is called as amniotic sac, right? When it is in the amniotic sac, what happens? It keeps on floating on the amniotic fluid, and this fluid prevents the prevents from baby from the shock, right? So some of the cells are taken out, some of the fluid is taken out with the help of injections. About 20 to 30 ml of fluid is taken out, and that fluid will be having cells of the fetus, and those cells of the fetus are taken out, they are cultured, they are stained, and with the help of chromosome analysis, we get to know whether the child is having congenital disease or not. If the child is having congenital disease, right, so patient or the person can go for, a couple can go for the medical termination, that is a medical termination of pregnancy, right? So these students all are the uses, or these are the certain tests. So in this question you have to write about what do you mean by medical termination of pregnancy, right? You have to explain why we go for medical termination. Secondly, you need to talk about, as the question is saying that how this medical termination are legally permitted. You have to talk about that. So you have to talk about both the acts. The first act was a Medical Termination of Pregnancy Act that was passed in 1971, right? And second is a Prenatal Diagnostic Test Act, right? And one thing you need to remember, this was passed in 1994, students, right? Later on, so many amendments was done in 2003, right? You have to talk about both the acts, right? Next, you need to talk about why this people go for, and the people go for this just because the child may be having some kind of congenital disease, or why they go for medical termination because of some problems that are related to the rape, or that are related to the when there is failure of contraception, right? These are the two things, right? And female, you have to explain about the female feticides and what are the methods, that is amniocentesis and sonography. Depending upon the marking of the question, if the question is of 3 marks or 2 marks, you can just write these two procedures under name, that's it. You know, you don't need to explain. When this question will comes in the 5 marks or the 5 marks question, then you have to explain what do you mean by amniocentesis, what do you mean by sonography. Right, students? So this was our first question, that was a medical termination of pregnancy and under what condition the MTP are legally permitted. Right? To talk about, this was our first question. Second we have is a describe the techniques which is used for sex determination in fetus. Right? Very important. This is also very important. We have already discussed this in the first question itself, but it was a very small discussion. Right? So, describe the techniques which is used for sex determination in the fetus. Now the students' question, we have to understand what do you mean by the question. It is a sex determination of fetus. Now, if we talk about the sex of a fetus, either the boy, either it will be the boy or the female or the girl. So, if it is a boy, right, it will be having XY chromosome, right? If it is a girl, it will be having XX chromosome, right? So we have to detect whether the fetus is having these two chromosomes, XY chromosome along with autosome, or it is having XX chromosome. Depending upon that, we can get to know whether the child is boy or a girl, right? We need to check that, right? So there are so many methods by which we can check out whether a child is female or a child is a male, right? The first basic procedure is the amniocentesis. The first procedure is the amniocentesis. Amniocentesis, right, students? And second procedure we have is the chorionic villi, or you can write villus sampling. Right. So students, no need to cram all those things, right? First we have amniocentesis. We have already talked about that we have to know whether the child is having XX chromosome or whether the child will be having XY chromosome, right? So what we have to do, we have to take out the cells of the child, right? There are two procedures by which we can take out the cells of the child. One is the amniocentesis. Amniocentesis. As the name indicates, amniocentesis. That means amnion and centosis. That is the amniotic fluid along with those cells, and centosis is to check. Now in this case what happens, we take out the cells. We take out the cells from amniotic fluid. Right? Amniotic fluid is taken out, and this amniotic fluid has cells. What? They have a cells of fetus. Or sometimes it is also written as FOETUS. Right? You can find both the spellings. This is the amniotic fluid is taken out, and there are cells of the fetus that has the cells of fetus. Now, now the question arises, how can we go for such procedure, and what is the exact time and what is the actual time by which we can go for this particular procedure? Like it says, from 12 weeks to the 20th week, it is safe for the amniocentesis. Before that, it is unsafe because amniotic fluid is very less at that particular time, right? So amniotic fluid is taken out, and then that will be having the cells of fetus, right? So students, come at this side. The first thing you need to explain, students, that what is the time of diagnosis, what are the things that fetus or it can be checked up to the 12 to 20 weeks, up to 12 to 20 weeks of gestations. Right, up to 12 to 20 weeks of gestation, we can go for this particular procedure. And in this case, what we do, a fine needle is inserted into the womb or into the uterus, right? Fine needle is inserted into uterus, right? A fine needle is inserted into the uterus, and fine needle is inserted, and around 20 to 30 ml of fluid is taken out, right? 20 to 30 ml of fluid. 20 to 30 ml of amniotic fluid is taken out, and that 20 to 30 ml of fluid, in which after the centrifugation the cells are removed or the cells are cultured, right? So cells are removed by centrifugation. Centrifugation, we all know centrifugation process, and the cells are removed. After the cells are removed, we culture those cells. We culture those cells. After the culturing of those cells, what we do, we go for one procedure, that is a chromosomal mapping, or to get to know with we can get to know whether the child is having some disease or not, or we can get to know what is the sex of a child. So next is a karyotyping. Karyotyping. Right. This is a very long procedure, karyotyping. Right. So actually what we require is 20 to 30 ml or 30 to 30 weeks of gestation. In between it is safe, right? Second, a fine needle is inserted into the uterus, 20 to 30 ml of fluid is taken out, and the cells are removed and they are cultured, and the karyotyping is done, and we get to know what is the sex of an individual, right? Now, one more thing I would like to tell you, in addition to this, that we are not restricted to only answer. If this question is for 5 marks, we can add some more things into it. If you talk about the amniotic fluid has some cells, right? And those cells of amniotic fluid are rich in stem cells. Right, students, they are rich in stem cells, they are rich in epidermis stem cell, they are rich in neural stem cells, they are rich in mesodermal stem cells, and they are rich in hematopoietic stem cell. Students, this is very important. And how we can use these stem cells? With the use of these stem cells, what we can do, we can culture the artificial heart, we can make the artificial liver cell, we can make the different arteries, we can make so many things out of it when the child is born with a congenital disease. So this type of cells, these amniotic sac cells or the fetal cells which are having this property, that is a stem cells, these can be used for curing those congenital diseases, right? So you can add on such things. So let me write that. That will be additional knowledge to you, right? Very important, students, very important as per your NEET is concerned, right? So the stem, this amniotic cells of amniotic fluid, cells of amniotic fluid are rich in, they are rich in what? They are rich in stem cells, and those stem cells are, first of all, neural stem cell. Neural stem cells, right? Neural stem cells means we can make neurons out of it, right? So there are mesenchymal stem cells. They are rich source of mesenchymal stem cells. One very important part, that is they are rich source of hematopoietic stem cell. Hematopoietic stem cell, right? So there are so many stem cells they have. I have already talked about the three. So these are the cells of amniotic fluids are rich in neural stem cells, mesenchymal stem cells, and hematopoietic stem cells. Now students, these stem cells are important when the child is born with some kind of congenital diseases or metabolic disorders. Students, is this visible to you? Congenital and metabolic disorders. Right, very important, students. Neural stem cells, mesenchymal stem cells, hematopoietic stem cells, right? Students, come at this side again, right? We have already discussed the amniocentesis, right? Now we will be discussing another topic, that is a chorionic villus sampling. Chorionic villus sampling is same as that of amniocentesis. The one basic thing in this case also we need to follow is that what we need exactly? We need a cells of a fetus. So while taking out the villi of the chorion, the membrane, the outermost membrane, we take out the chorionic cells, right? And that has a cells of fetus, and by culturing those cells, by staining those cells, by karyotyping those cells, we can get to know what is the actual sex of an individual, right? So to talk about this, the question is to describe the technique which is used for the sex determination in fetus. We can write these two, one is the amniocentesis and another is a chorionic villus sampling, right students? So next we have another question, describe the three methods in which fertilization of human ovum by sperm can be prevented. Students, in the lectures we have discussed about the certain methods of contraceptions, right? We have already discussed some natural methods are there, right? Those natural methods could be, there are so many natural methods, right? There are so many natural methods are there. Other than that, certain barrier methods are there, right? This is for those students those students who do not have revised all those things, that what do you mean by this fertilization or the sperm entry into the vagina. Now, we have to tell about the 3 manners in which the fertilization means combining of the male gamete and the female gamete, right students, of the human ovum by the sperm can be prevented, right? So one thing we need to keep in mind, we have to stop the meeting of the sperm and the ova. This is the way we can prevent the fertilization, right? As we have talked about in the lectures, we have already talked about certain methods, certain natural methods, right? Like coitus interruptus, there are so many natural methods that I don't think so that is a need to discuss over here. One thing which we have to discuss over here are the barrier methods, right? We will be discussing about the barrier methods, right? So students, to talk about the barrier methods are those methods which prevent the meeting of sperm and the ova together, right students? So this is barrier method, right? Right? In this case, prevent meeting of sperm or the male gamete with female gamete or the ovum, right? And this is our barrier method, right? To talk about the barrier methods, there are so many barrier methods are there. First, the basic barrier method that we all know is a condom. So students, come at this side. First barrier method is the condom, and these are used by male, right? These are used by male, and they are made up of latex, right? And that a male wear such a condom over the male reproductive part, and so that the sperm cannot be inserted into the female vagina, right? Second we have is the right. Second we have is a fem shield, right? Fem shield, right students? And this fem shield is also called as a female condom, right students? This is a female condom which is made up, this basically covers what? It covers the external genitalia, right? When it covers the external genitalia, now what will happen? When the sperm will enter, it will not be able to penetrate inside because it is covered by one fem shield. That is also called as a female condom. If we talk about that, it is made up of one chemical which is called as a polyurethane. Polyurethane. They are made up of one thing, that is a polyurethane, right? There are so many other methods are also there, like diaphragm. Except condoms, all are used by the females, right? The fem shield, which is also called as a female condom. The third is we have is a diaphragm. Right? If we talk about, it is made up of one rubber, right? Right? To talk about the structure, it look like this. Right? Right? This is made up of rubber, and other than that, this is a ring, and this is made up of metal. This is a metal ring, right? And it is placed over the vagina. placed over the vagina. Right, students? It is like this is a rubber. This white part is a rubber, students. And this is a metal ring. It is placed over the vagina so that the sperm will not be able to penetrate into the women's reproductive part. Right? Like, to talk about all those things, there are so many things are there. One is the diaphragm. We have already discussed the diaphragm. We have discussed the condoms. That is the male condom. We have discussed the female condom. Along with female condom and diaphragm, certain chemicals are also used, and those are spermicides. Like spermicides are used along with diaphragm, along with spermicides are used along with diaphragm. Now the question arises, students, what do you mean by spermicide? Spermicide means sperm and side means sperm are killed. These are those chemicals which kills the sperm. So can you give one example of one spermicide? Hope my students know about it. That is that I have discussed in the classes. That is a nonoxynol 9. Nonoxynol 9. Nonoxynol 9 is a spermicide which is used along with the diaphragm and the female condom. So it increases the efficiency of the female contraception, like contraceptive methods, right? They are used along with the diaphragm and they are used along with the this female condoms, right? Other than that, we have some other, that is a vault cap. Similarly, like that of a diaphragm, vault caps are also there, right? Fourth we have is the vault caps. Fourth, vault cap. This is also the same, made up of the same chemical, that is a polyurethane. Polyurethane. It is placed over the vagina and used by the females, right? Used by females. Right? They are used by the females. Now, what happens? It looks like this only. Right? Along with the vault cap, the spermicides are also used. Spermicides along with vault cap, right, increases its efficiency. Right. So what will happen? Whenever the sperm will enter into the vagina, first of all, it will be killed by the spermicides, right? As it will be killed by the spermicides, some of the sperms which will remain, they will not be able to enter because the whole reproductive system is covered by one particular thing that is called as a vault cap. Right, students? So with this, we have discussed about the three manners by which the fertilization can occur. So students, come at the side. So question was, describe the three manners in which the fertilization of human ovum by the sperm can be prevented, right? So in this we have discussed about the certain barrier methods, right? So in this question you have to write the whole, you have to give examples of everything. You start with the burial method, what are burial method, how it is used and what is the function of burial methods. You will give some certain example of burial methods. Students, along with this burial methods, there are so many other methods are also there. Because here the fertilization has mentioned, right? So we cannot write the intrauterine devices. Sometimes the intrauterine devices can also be mentioned over here. Some intrauterine devices are also there, such as a copper T, right? Copper T keep on releasing the copper ions, right? Due to which the fertilization cannot occur. One thing you need to remember in this case is that fertilization occurs in the fallopian tube, right students? The sperm will enter into the vagina, they will reach up to the fallopian tube where will be the fertilization will occur. Next question to talk about is, so students, this was all about this class. In the next class, we will be discussing some more questions related to this. Till then, students, revise all this. We will be coming in the next class. Till then, take care. Bye-bye, students. Thank you. Now, students, now it's the time for the third question. That is, describe the 3 manners in which fertilization of human ovum by the sperm can be prevented. Now, students, one important thing, that is a fertilization. Students, you need to know that what do you mean by fertilization. Fertilization is the meeting of the female gamete and the male gamete. When these 2 gametes, they will come close to each other, what they will lead to? They will lead to the fertilization. Now, the question is asking that we have to talk about the 3 manners in which the fertilization of human ovum by the sperms can be prevented, right? So, what we have to do, we have to talk about those methods where the sperm entry can be prevented, right? So, there are 3 methods. The first method is the barrier method, right? First method which we will be talking about in this is the barrier method. Second, in this case, could be certain chemical methods. Right students? And third, we have certain intrauterine devices. Right. There are so many other methods also which you have already studied in my classes. There are some natural methods are also there, like biological rhythm method, like coitus interruptus method, right? So many other methods, natural methods are also there, and that natural method can also be mentioned over here. But I'm basically focusing on these 3 just because these 3 are very important and students consider these 3 in a typical, like they think these are the typical, so I'm taking all like these 3. First, as the name indicates, the barrier method, right? This barrier method. Barrier methods are those methods in which what happens is that sperm is not entered into the vagina, right? Because of the presence of certain barrier, right? Whenever those barriers are there, the sperm will come, it will not be able to enter into the vagina, it will not be able to enter into the fallopian tube where the fertilization can occur, right? So this comes under the barrier method. Next, we talk about another thing, that is a chemical method. Chemical methods means we are using certain chemicals so that the sperm entry could be prevented. And those chemicals could be certain spermicides that are applied to the female reproductive part, so that whenever the sperm will come, the sperm, when it will come in contact with the spermicide, it will die itself. It will not be able to penetrate into the fallopian tube. Now, where there the fertilization can occur, right? So, there will be no fertilization in this case also. Next, we have is intrauterine devices. Intra means inside, uterine means inside the uterus, and devices means a certain methods or certain mechanical methods are there which are inserted into the uterus so that the sperm entry could be prevented. And sometime to exactly tell about the intrauterine devices, these uterine devices also prevent the implantation, right? So firstly, I'll be taking one method, that is a barrier method. So students, come at this side. Barrier methods. To talk about the barrier method, the first method, that is the barrier method, it prevents the entry of the sperm into the uterus. So, prevent the entry of sperm into uterus, right? One more thing you need to remember is the fallopian tube is a place where fertilization occurs. Right, students? Fallopian tube is a place for fertilization. Right. So, this is a fallopian tube where the fertilization will occur. Now, in the barrier method, what we'll do, we will not allow the entry of the sperm into the vagina. So, in this case, what exactly happens is, there are certain barrier methods. We all know about, there are some basic methods are there, like we all have heard about. First is a condom. First is a condom. This is used by male. Some female condoms are also there. We are talking about the normal latex condom. So, it is made up of latex, right? It is made up of latex, right? Used by males, right? And it prevents the entry of the sperm into the fallopian tube, right? It is used by the male, and what they do, a male wear this condom over their testes, over their penis, and so that whenever the sperm will be deposited on the condom itself, right? So, wear over penis, right? So, the sperm will be deposited on the condom itself, and it is made up of latex. So, first we have already talked about, that is a condom. And another than the male condom, we have one more condom, that is a female condom, which is also called as a fem shield, students. Fem shield. Second we have is the femine, right? Fem shield. Fem shield, right? And these are called as a female condoms. Female condoms, right? These are those condoms which are used by. So, students, while attempting this, what you have to do, you have to write certain things about the barrier methods, certain things about the chemical methods, and something about the intrauterine devices. So, first we are discussing this barrier method. So, I am giving you 2 to 3 examples of each. So, second was this condom, and second is a fem shield, which is also called as a female condoms, and it is made up of one chemical which is called as polyurethane. Made up of polyurethane, right? And this is used by the female, as the name indicates, itself is a female condom. So, female wear this over the vagina or external genitalia. It covers the whole vagina and external genitalia. Right? And these are the methods, not only the method of contraception, other than that, these are also the methods by which we can prevent sexually transmitted disease, that is STD, right? So, these are wear over the vagina and the external genitalia, and the sperms, while copulation, sperms are deposited over the fem shield, and fem shield will protect the entry of the sperm into the vagina. Third, we have one more, that is a diaphragm, which is also used by the female. Is the diaphragm. This is also used by female students, right? Used by females, right? And diaphragm basically look like this. They have this metal or plastic lining over this, lining like this. This is a double lining. and this is closely fitted into the vagina. Over the external genitalia, it is closely fitted over there. So it prevents the entry of sperm into the vagina, and this is made up of rubber. This outermost is made up of rubber. Right? Another, we have this ring, which is made up of metal, or called as a metal ring. We don't need to go in detail because we are answering a CBSE question, right? So third, we have discussed about the diaphragm. If we talk about the diaphragm, and the third we have, fourth we have a vault caps. Vault caps. Fourth, we have. There are so many methods, so many barrier methods are there, but I have compiled only few. Vault caps. Vault caps are also made up of polyurethane. Right? They are also made up of polyurethane. Right? It's like this. It covers the cervix, right? It covers the cervix so that the sperm will not be able to enter into the vagina. The third is fourth is a vault cap. Other than that, some vaginal plugs are also there. So vaginal plugs look like this. Right? So they cover the whole cervix and prevent the entry. Right? So these barrier methods, as they prevent the entry of the sperm into the vagina, along with that, if it is used along with the spermicide, in case of those contraceptive methods which are used by the female, if those female uses this chemical methods along with this contraception method, that is, if they use spermicide along with this, spermicide, if they will use this spermicide along with this vault cap, or they will use this spermicide along with this diaphragm, this increases the efficiency, right? Increases the efficiency of barriers. It increases the efficiency of barriers when they are used with a spermicide. Like nonoxynol 9, this is one of the spermicides which is there in so many sponge which are available in the market in the name of today. Right? So first was a barrier method. Next, students come at this side. Next we have, as we have discussed the barrier methods, now we will be discussing the chemical methods. As the name indicates, chemical methods, that means certain chemicals are used to prevent the entry. So students come at this side again. So we have chemical methods. Chemical methods are those chemicals which act as a spermicides and that a female applies into the vagina. So whenever the sperm will enter into the vagina, now what will happen as the sperm has entered, the vaginal covering will be having those spermicide layer. When the sperm will come in contact with those spermicide layer, the sperm will be killed, right? So next we have is the chemical layer, or the chemical method, right? The second method, right? As the name indicates, chemical method, students. Chemical methods means chemicals used to prevent, chemicals are used to prevent entry of sperm into vagina. Right? Chemicals are used to prevent the entry of sperm into the vagina. Now, the case is different. We use some chemicals, that is spermicide. Spermicides are used. So students, like how to learn all those things? It's very easy, no need to cram all those things because science is all about the day-to-day life. So spermicide, sperm, side means sperm, kill means to kill the sperm, right? Chemical used to kill the sperm. chemicals used to kill the sperms. And can you explain some spermicide? Yes, it is nonoxynol, right? And it is available in the trade name of today. Available by the trade name of today, right? And this today has sponge-like structure. They have some sponge-like structure that has this nonoxynol, that is a spermicide over there. When a female applies that sponge into their vagina, all the spermicides will be released into the female vagina. So whenever the sperm will enter, now what will happen? The sperm will kill and there will be no fertilization, right? So students, this was the chemical methods. Next we have a third method, that is intrauterine devices. Intrauterine devices, students. Intra means inside, uterine means inside the uterus, and devices means those mechanical things which are inserted into the female part so that there is no fertilization, there is no implantation, right? So we will be discussing, because it's a 3 to 5 marks question, so we don't need to explain that much. So the third we have is the intrauterine devices. Means these are those devices or mechanical devices which are inserted into the female part so that there will be no fertilization and implantation. Now students, I have used one another term, that is implantation. That means whenever the sperm or ova will fertilize together, there will be the formation of the zygote, and that zygote, what will happen? They will come into the uterus, and where it will implant itself into the endometrium, right? So what we have to do, we have to control those things. We have to control the copulation, we have to control the implantation. No, so we have one devices, that is the intrauterine devices, and one example of intrauterine devices is the copper T. Right? Copper T, it is also available in the form of multi-load 345, and the copper T, as the name indicates, copper means what is it? It is a metal, right? And what is the function of this copper T? T, just because its structure look like a T, it has something structure like this. One thread is attached over like this, so this is how a copper T look like, very small structure. And the female, like a doctor, they insert this copper T into the female vagina so that whenever the sperm will come in contact with these copper Ts, they will die. Now the question arises, how they die? What actually happens, students, in this case? These copper T releases calcium ion, they releases copper ion in a very frequent time. Like every day it releases around 50 microgram per day. What they do, they release copper ion. It releases copper ion, and the frequency and the concentration of it is 50 microgram per day. Right? Right? When this copper ion are released, now what will happen? As a copper ion are released into the vagina, now what will happen? The cytokines, our immune system will evoke, and the cytokines will come over there, they will block that particular area. So whenever the sperm will enter into the vagina, they will be killed by or they will be blocked by those cytokines. Now what will happen? Because of the presence of copper ion, what will happen? Cytokines will be released, and these are the immune system cells we have already discussed in the human health and disease. Hope you remember, students, this as well. Cytokines, these are the immune cells which are released by the immune system. So these cytokines, they block the entry of the sperm. So what they do, they block the entry of sperm. Right, they will block the entry of the sperm. Now, as a copper is releasing at 50 microgram per day. So this copper T has specific life, and its specific life is around 2 to 5 years. Right? Its life is around 2 to 5 years. So that some another are also there, that is a multi-load 345. So many properties are available which are called as intrauterine devices, right? The main thing is that it prevents the fertilization, students, right? So students, this was the third question and we have discussed all the three aspects, right? Now we will be discussing the next question, that is a question number 4. So the question here says, suggest some methods to assist infertile couple to have children. Infertile couples to have children. So students, one thing you need to learn, that is what do you mean by infertile couples, right? We have discussed in the class that what do you mean by infertility. Infertility is an inability of an organism to reproduce. That can be a male infertility and that can be a female infertility. In both the cases, what actually happens is, sometimes what happens, the sperms are not produced, sometimes the ova or the ovum is not produced or it is immature, right? In some cases, what happens, the female body is unable to carry that baby. In that particular cases, the couple go for assisted reproductive technologies, right? So there are so many methods that a couple can adopt and these are called as assisted reproductive technologies. Assisted reproductive technology. Right. Next is assisted reproductive technology. These are the methods which a couple can adopt if they are infertile, right? Now the question arises, why the couple are called infertile? This is just because of some reasons. When they try to reproduce up to the 2 years, right? And even after that, when they are unable to reproduce, they are called as infertile couple, right? So assisted reproductive technologies are there. First, we have one is IVF, right? IVF, as the name indicates, IVF, that is in vitro fertilization. So students, you have to explain some subtopics that comes under the assisted reproductive technology. The first is the in vitro fertilization. So students, come at this side again. So as the name indicates, in vitro fertilization means fertilization outside the body is called as in vitro fertilization. Whenever we do fertilization outside the body in the petri plate or in the laboratory, this is called as an in vitro fertilization, right? So what actually happens in vitro fertilization is that, right, suppose the fertilization has to occur, right? Suppose in some cases what happens, the female gamete is not produced, right? In some cases, the male gametes are not produced in case of some infertile couples. So what we do, we take out those gametes from the body, or we take the gametes from the donor's body, right? We take out both the gametes, right? We fertilize them inside the laboratory in the petri plate, and that is called as in vitro fertilization, right? So students, so as the name indicates, in vitro fertilization means fertilization outside the body of female fertilization outside the body of female in petri plate or in any devices where fertilization can occur. This is called as in vitro fertilization, right? In this case, why we go for this in vitro fertilization? When female is not able to produce mature ovum, which is also called as a Graafian follicle, students. Out of Graafian follicle, the ovum come out and produce mature egg, or you can say ovum, or when in case the sperms are not produced by the male, or the male cannot able to produce the sperms, right? In this case, we go for IVF technologies. There are certain other ways also, like suppose a female is unable, the female fallopian tube is blocked somewhere, right? The fallopian tube is that place where fertilization occurs, right? So when there will be no fertilization, because fallopian tube is not there, what the doctor will do, they will take out the egg of the female and they will take the sperm of the female, they will fertilize them outside the body in the Petri plate, so that will be called as in vitro fertilization. So this was our first, that is the in vitro fertilization. Next we have is intracytoplasmic sperm injection. Intracytoplasmic sperm injection. Next, assisted reproductive technology method. Second we have intracytoplasmic. First was IVF, second is this intracytoplasmic sperm injection. Right. As the name indicates, intracytoplasmic sperm injection. That means the sperm is taken out from the female, from the male body, and that sperm is injected into the cytoplasm, right? Suppose, I'm just giving you one rough diagram. Suppose this is a human egg, the female egg. Now what will do? A sperm is injected, right? And sperm nucleus is injected into the cytoplasm, and so that the fertilization can occur. This we go for, this is an egg and this is a sperm, right? This is a case of IVF only, that we can do all these things outside the human body also, or it can be done into the human body also, where the sperm is injected into the female vagina, and those couple goes for intracytoplasmic sperm injection where the male is unable to produce the sperm, right? When the male is infertile, right? Or sometime what happens, the motility of sperm is affected, or sperm of the male is immotile. If sperm is immotile, right, then we go for this intracytoplasmic sperm injection. Because why motility is important? Motility is important because this sperm has to travel from vagina to the fallopian tube, right, with the help of the flagella which it has. So it reaches up to the fallopian tube where fertilization can occur, right? So in this case, couple can go for this intracytoplasmic sperm injection, or in the second case, when the sperm is not functional. Not functional. In this case also, couple can go for intracytoplasmic sperm injection. So this may be asked in the this is ICSI, intracytoplasmic sperm injection, right? Next reproductive technology we have is GIFT. GIFT. Gift is given, right? So you can consider it as a G means gamete, intrafallopian, intrafallopian transfer. Right. As the name indicates, gift, that is a gamete intrafallopian transfer. In this, what happens exactly is, sometime this couple go for this procedure, that is a gift. The gamete is implanted into the fallopian tube, right? And this basically, females goes for this because whenever the female is unable to produce the ova, the egg is not produced, and the egg is not there in a fallopian tube. So when the sperm will come over there, there will be no fertilization. So what will happen? There will be no fertilization, there will be no pregnancy. So in this case, what we go for with the gametes, the female gametes is implanted into the fallopian tube, right? Female gamete, or you can say egg, right? Female gamete or egg is implanted, or you can say inserted into fallopian tube. And when they are inserted into the fallopian tube, the fertilization can occur, right? And these fertilized egg can be from the donor organism, because the female is not able to produce the egg. So there are chances that the egg is not good, or there will be no fertilization, or there will be some congenital diseases in the body. So in this case, the couple go for one procedure, that is GIFT, and the female gametes they take from the donor organism, right? Next, another procedure along with GIFT, we have ZIFT, ZIFT. Fourth we have is ZIFT. Earlier it was GIFT, so I have mentioned both, that is now it is ZIFT. ZIFT, as the name indicates, Z means zygote, intrafallopian transfer, right? Earlier it was gamete intrafallopian transfer, now it is zygote intrafallopian transfer. Zygote intrafallopian transfer. So what happens in this case? Zygote, now the question arises, what do you mean by zygote? Whenever the female gamete or male gamete, they combine together, the first stage which is formed is called as the zygote. And that zygote, they will transport itself into the uterus where the implantation will occur, right? Sometimes what happens, the female is unable to produce the egg, right? Sometimes what also happens, this can also happen that the sperm is not produced. So in this case, what the couple go for IVF, that is in vitro fertilization. After in vitro fertilization, the whole egg is transplanted into the whole zygote is transplanted into the uterus, and where the implantation can occur, right? In this case, either their fertilized egg is entered into the, they are supposed to enter into the fallopian tube, or they are made to enter into the uterus, depending upon which stage it is, right? Up to 8-cell stage, right? 8-cell blastomere stage, it is in the fallopian tube. After the 8-cell stage, it is implanted into the uterus or in the endometrium, right, students? So with the help of certain hormonal injection, we go for this procedure, that is a ZIFT, because we have to make the female body or the female body so that there should be an implantation. So implantation can properly occur. The endometrium should be properly made. Next, a couple can go for, in this case, is the surrogacy. Surrogacy. Surrogacy is banned in India. Somehow, some laws are about to ban in India. It's not actually banned. And surrogacy means we look for a female who can carry the baby for 9 months. According to law, our law says a patient or a couple can go for the surrogacy only when the female is infertile, or in some cases when female is unable to carry that baby for 9 months. In that cases, the couple can go for surrogacy, where they look for a female who can carry their zygote or their baby for 9 months, and later on when they will give birth, they take that baby from that particular lady. So it is very much prevalent in India, but as surrogate mothers are very cheap in India, so people from the outside country, they come to India for the surrogate mother and for the surrogacy, because they don't want, or they cannot carry the baby for the 9 months, because they feel that the carrying a baby brings pain into their body. So this is also a surrogacy. So students, this was the methods. Students, come at this side. So we were talking about the assisted reproductive technologies, so with the help of which a couple can have a children. The first is the IVF, that is the in vitro fertilization. Students, come at this side again. Second, we have discussed about the intracytoplasmic sperm injection, which I have removed from here. And the third was the GIFT, fourth is the ZIFT, and fifth one is the surrogacy. Right, students? So students, note down this.